Hello, I'm Deborah Gordon. I'm a professor at Stanford, and I'd like to talk to you today about the ecology and evolution of collective behavior in harvester ants. I study collective behavior and how systems are regulated without central control. An ant colony is a great example of a system without central control because there's nobody in charge. A colony consists of sterile female workers, the ones you see walking around, and although there are one or more reproductive females, and we call them queens, in fact, they're not in charge. They don't tell anybody what to do. They just lay the eggs. I've been studying harvester ants at this site in southeastern Arizona now for 28 years. The species is called Pogonomyrmex barbatus. And they're called harvester ants because they collect seeds and they eat seeds. This shows the nest of a colony, a mature colony. The ants leave the nest entrance and they travel on trails that might be cleared or might not be for up to 20 meters, collect seeds and bring them back to the nest, and they store them inside the nest. And this behavior, the foraging activity of a colony, as well as all of its behavior, is regulated by local interactions among ants. This shows ants in a lab arena, moving around, interacting with each other. The arena is connected by tubes to two other arenas. And this shows the network created by all the ants moving around and interacting. And when we look at this network and we look to see which ant is interacting with which one, we can see that some ants interact more than others. They become hubs. But what we learned from this study is that ants become hubs because they go to the hotspot where there's a lot of interaction. It's not a characteristic of particular ants. And so in this sense, the network of interactions that these ants are using is a distributed network. It doesn't matter which ant meets which. Ants participate in the network as a function of where they happen to be. Harvester ants use interaction networks consisting mostly of brief interactions when one ant touches antennae with another. Most ants operate mostly by smell. Ants don't see very well, and they smell with their antennae. And when one ant touches another, it can tell if the other ant belongs to the same colony and what task it's been doing. And these interaction networks then carry no other message. What the ants are detecting when they smell each other are the cuticular hydrocarbons of the other ants. Ants, like many insects, are covered with a layer of grease, long-chain fatty acids, that carry the colony-specific odor. And in this species, at least, they also indicate what task the other ant has been doing. And that's because the conditions under which an ant is working change the chemistry of its cuticular hydrocarbons. When an ant is outside foraging in the hot sun, the chemistry of its cuticular hydrocarbons changes. And so a forager smells different from an ant working inside the nest. With my colleague, Mike Green, we've been doing experiments to learn how ants are using interactions. So the experiments consist of using little glass beads or other small objects coated with an extract of cuticular hydrocarbons. And by doing this, we can manipulate the interaction rate. And this is how we learned that there's no message other than the particular kind of cuticular hydrocarbon and the rate of interaction, because ants will respond to the rate of interaction with a little glass bead in the same way that they would respond to interactions with other ants. So harvester ant colonies have to solve ecological challenges like any other species. And a harvester ant colony's biggest problems are, first of all, conserving water, because they live in the desert and water is scarce, and then competition with neighbors of the same species. The seeds that they eat are sparsely distributed by wind and flooding. They're just scattered at random. And so harvester ants don't use pheromone trails. There's no need to recruit because one ant can bring back a seed by itself, and the seeds don't occur in patches. So if you're an ant and you find a seed, that doesn't mean there's going to be any more seeds nearby. There's no point going back to get anybody else to recruit to the seeds. Now, foraging costs water, which comes from seeds. An ant out foraging in the hot sun, searching for seeds, loses water in the hot desert air. And they get their water by metabolizing the fats out of the seeds that they eat. So basically, a colony has to spend water to get water. They have to spend water by sending ants out to forage in order to get water out of the seeds that they collect. So the colony faces a trade-off. Too much time spent foraging, they are losing water. But if they spend too little time foraging, they actually lose foraging area to neighbors because all the neighbors are out searching for seeds in the same places. So the colony manages this trade-off through a network of interactions among foragers as they come in and out of the nest. So the question is, how does a colony regulate its foraging activity? How does an ant decide, okay, it's time to go out and search? What gets the foragers out? What regulates how many foragers are out right now? First of all, it's not a simple consequence of how many ants there are, because the same colony will send out different numbers of ants within a day, from day to day, and there's a lot of difference from one day to another in how many ants are foraging in all the colonies, depending on how hot and dry it is. So there is moment-to-moment regulation of how many ants are going out to forage. This is how it works. When a forager goes out, depends on the rate at which foragers return with food. So an outgoing forager doesn't leave the nest unless it has enough interactions in the form of antennal contact with returning foragers with food. This makes sense for the colony because it leads to higher foraging effort when more food is available. The more food is out there, the less time foragers spend searching, foragers return sooner, and they send out more foragers to forage. So it's a kind of positive feedback. So we're trying to understand exactly how this works. And to do that, we need to know a little bit more about what's going on in order to try to measure the rate at which they interact and how that determines the flow of foragers. So to learn more about that, I dug up the nest entrances of some colonies to see exactly how they're interacting inside the nest. This shows the nest entrance of one colony, and you can see, first of all, that there were 2 nest entrances to begin with. And then when I dug it up, I found 2 tunnels, and those are indicated with flags, that lead to an entrance chamber. It's about 5 centimeters from the surface where the nest entrances were leading in. And in that chamber, there was another tunnel leading deeper into the nest, and I found that seeds were piled there at the end of the foraging period. We were able to use a videoscope to film the interactions of ants inside the nest. And you can see here, this is a film made with a videoscope inside the nest. The foragers are coming in, and they're engaging in antennal contact with the outgoing foragers. There's a millet seed in the back there that you can see. So those antennal contacts, like those, are what determine the rate at which foragers leave. And one of the things that's so fascinating to me about this is how messy this process is. It's not as if the rate of interaction is very regular, and each ant doesn't respond deterministically. If you've watched ants at all, you've learned that an ant doesn't really do the same thing every time in the same circumstances, but with very messy input and very messy output, the colony regulates its foraging in a very predictable way. So here's another way to illustrate what's happening. In this story, the red ants are the returning foragers and the blue ants are the outgoing foragers. And so the returning foragers come in with food. They don't really forage for apples, but they come in with seeds. And they meet the outgoing foragers in the tunnel. And so as the foragers come into the tunnel, they pass the outgoing foragers and they engage in antennal contact. And eventually, the returning forager puts down its food, which is taken by another ant, shown here in green because it's not part of this story, that comes up from the deeper nest, gets the food, and brings it back down. And then if you look at that red ant that just put down the food, it becomes a blue ant. It becomes an outgoing forager, and it's waiting for contact with returning foragers to decide whether to leave the nest and go out to forage. So we'd like to understand exactly what is the algorithm that links the rates of outgoing and returning foragers. And to learn about this, we've been doing experiments where we decrease the rate of return for only a few minutes. So this shows forager rate in minutes, and we take away the returning foragers by using our most important piece of field equipment, the plastic box. So we're out there picking up the ants as they're coming back to the nest with food, with seeds. We put them in the box, and that causes a temporary decrease in the rate at which foragers are returning. And at the same time, we then see, after a lag, which is interesting, a decrease in the rate at which foragers leave the nest. And that decrease in the rate at which they leave the nest is larger than can be accounted for by the fact that there's lots of foragers stuck in the plastic box. So when the rate at which foragers goes in decreases, the rate at which foragers go out decreases. When a forager goes out, it depends on the rate at which foragers return with food, but not foragers returning without food. We did some experiments early on in which we took away the few ants that come back without food, and we found that it made no difference to the rate at which foragers are leaving. So that raises the question, do the inactive foragers, the outgoing foragers inside the nest, respond to interactions with the returning forager, with the food, or both? So here we are doing what we do. Here's Mike Green putting little things down into the ant nest. And here I am putting little things down into the ant nest. And the little things were small silica chips coated either with the cuticular hydrocarbons of foragers, oleic acid, which is a common component in seeds, so this corresponds to food, or both, or a blank control that really just had the solvent that we use to extract the cuticular hydrocarbons. And here are the results. It was only in response to the combination of forager hydrocarbons and food that foraging increased. This means that the ants need both the forager and the food to respond to the interaction in a way that stimulates foraging. And this is really interesting because the ants put down the food shortly after they come into the nest. So by using the odor, the combined odor of hydrocarbons and food, the ants are getting a more precise assessment of the rate at which they're actually coming in. So this is a very interesting refinement of the interaction network that allows them to regulate foraging more closely to the rate at which foragers are finding food and bringing it back in. Here's what the real data look like. This shows the rate of foragers going in and out of the nest in seconds. The whole thing is about 14 minutes. And the red line corresponds to the rate at which foragers are coming into the nest. The blue line corresponds to the rate at which foragers are leaving the nest. And the green bar shows the time over which we were removing returning foragers, collecting them up off the trail, and putting them in a box. So you can see that during the time that we're removing returning foragers, the red line, the rate at which foragers are coming in, goes way down because we're picking up the foragers. And at the same time, the rate at which foragers are leaving the nest goes down also because the ants inside the nest are not meeting the returning foragers. And then we stop collecting the returning foragers, so they start to go back, and the red line goes way up because we're not bothering them anymore, we're not interfering anymore, and so the foragers are continuing to return. And after a little lag, which is interesting, the rate at which they go out picks up again, and they start leaving the nest again in response to the returning foragers. Again, what's really interesting to me about this is how messy it is. You can see that the rate of forager return and the rate of outgoing foragers is not very... Again, what's interesting to me about this is how messy it is. You can see that the data are quite noisy, and yet the response is very predictable and strong. The internet uses an algorithm to regulate the flow of data that is very similar to the one that harvester ants are using to regulate the flow of foraging. And Balaji Prabhakar and I call this analogy Anternet. Data doesn't leave the source unless it gets a signal that the previous packet of data that left the source had the bandwidth to go on. So just as harvester ants are using the rate of return of successful foragers as a signal that it's worth it to go out and forage, so in the internet, this protocol called TCP/IP is ensuring that data doesn't leave the source unless it's worthwhile because there's bandwidth for it to go on. So especially in the early days of the internet, when it was very small and operating costs were very high, and it was really important not to lose any data, they set it up in this way using an algorithm very similar to the one that the harvester ants have evolved to regulate the flow of data. And of course, it's very interesting that ants are using an algorithm that looks like one that we recently invented, but this is only one of a handful of ant algorithms that we know about. And there are more than 12,000 species of ants. And the ants have had more than 130 million years to come up with interesting algorithms for data networks. So I think it's very likely that some of the other 12,000 species of ants are going to be using algorithms that we've never even thought of. So now we can ask how these interaction networks that regulate foraging are evolving in this population. In other words, we can ask whether there's natural selection going on right now on the regulation of foraging. Are the colonies that are making better foraging decisions showing higher reproductive success? That is, we could try to find out if natural selection is shaping the ongoing evolution of collective behavior in this population. Natural selection requires variation, heritability, and differences in reproductive success due to the trait that's variable and inherited. So first of all, variation. We find that there is variation in the regulation of foraging in this population of colonies. On dry days, some colonies reduce foraging. That is, colonies are different in how they manage this trade-off between spending water by going out to search and getting water in the seeds that they eat. Colonies of harvester ants live for 25 to 30 years, and they show consistent behavior year after year. A particular colony that forages less on dry days one year also forages less on dry days another year. So this is consistent variation among colonies that persists apparently as long as the colony lives. And we're trying to understand then, why do some colonies forage less than others? And we're doing this by thinking of the ants as neurons and drawing on models from computational neuroscience. So a neuron has to add up its electrical stimulation from other neurons to decide whether to fire. In other words, a neuron integrates its stimulus from other neurons to make a decision about whether to fire. And in the same way, or in an analogous way, an ant is adding up its interactions with other ants to decide whether to go out and forage. So what we're looking for here would be differences among colonies and how each ant integrates or adds up its interaction. Maybe in some colonies, each ant needs more interactions in order to be willing to go out and forage. And if so, a colony with ants like that in it is going to forage less because it's going to take more ants returning with food before the ants go out and forage. And so for a given amount of food, a colony in which each ant requires more interactions is going to do less foraging. So to think more about how this might work, let's think about how ants respond to interaction rate. And so here, I'll tell you a story about how I think it might work that's analogous to the way a neuron works. Let's imagine that each interaction stimulates some physiological response in an ant. And that response has a decay. So every time it experiences an interaction, if the next interaction comes before the last one has decayed, then eventually it would have enough interactions to push it over some activation threshold, which would then, for example, make it very likely to go out and forage. We think that this is what happens. From our experiments in which we can manipulate the rate of interaction by using beads coated with extracts of hydrocarbons, it seems that the period over which an ant is responding to interactions, the period over which an ant is accumulating its experience of interactions, is about 3 to 10 seconds. This is very analogous to the way that neurons work. So we're using integrate and forage models by analogy with integrate and fire models. Each ant is a leaky integrator of its interactions, and we're looking for differences among colonies in how leaky they are in this decay rate in an ant's response to interactions, to see whether we can find exactly how ants are different in different colonies that would add up to the very large differences that we see among colonies in how much they forage. So we're really trying to understand the source of the variation among colonies. And the next step will be to look for the genetic variation that might underlie this. Now, an important component of natural selection. A crucial component of natural selection is that variation in regulation of foraging is associated with variation in reproductive success. In order to find out about this, we needed a measure of reproductive success. Ants don't reproduce more ants, colonies make more colonies. And so it hasn't really been possible to measure the evolution of the collective behavior of social insect colonies because we've never really been able to measure reproductive success. How many colonies does another colony produce? But because of my long-term data in this population, we've been able to do this. The colony is the reproductive individual in a population of colonies. And so I want to tell you a little bit about how a colony reproduces, how colonies produce more colonies. This is a map of my study site. This is the map from 1993. And I have mapped this population every year since 1985. The map shows a road along the bottom, and every point in this map is the location of another colony. And every year I go out and find all the colonies that were there the year before, that are on the map from the year before. I take off the ones that have died, and I put the new ones on. And so in this way, I've been able to track these colonies for many years. This is how colonies reproduce. And this is a very common method of reproduction in many species of ants. So the colonies reproduce in an annual mating aggregation. In this species, it happens in the summer after the rains. All of the colonies in the population send out winged reproductive males and females. You can always tell the males from the females in ants because the males have smaller heads. And that's because they don't live long enough to consume much food. They don't really need their head for what they're going to do. And so the males always have smaller heads than the females. Then all of the winged males and females fly to one spot where they mate. And this shows them mating. This shows ants mating. Here, this female who still has her wings is mating with that male. And there's another male on top waiting his turn. It turns out in this population, because of an interesting genetic system, that each female has to mate more than once in order to produce a viable colony that can then reproduce. So the females mate many times. Then afterwards, the males all die. And then the newly mated queens fly off, apparently at random. They can shrug off their wings, and then they dig a hole. And this is the nest of a newly mated queen dug the same day. They go down about 18 inches. And the queen will stay there for the rest of her life, continuing to reproduce all of the workers, the reproductive females, and the males using the sperm from that original mating session. This is a colony 5 years later. It gets to be about 1 meter across. The ants cover it with pebbles that they use to store the cuticular hydrocarbons that marks their colony. And once a colony gets to be about 5 years old, it doesn't get any larger, and that's when it begins to reproduce. That is, that's when the colony makes winged reproductive females and males to go out to that year's mating flight. So the story that I want to tell you is a story about mothers and daughters, and so I'm going to show you the people who worked on it with their daughters. This is Krista Ingram with her daughters, Anna Pilko with her daughter, and there I am with my daughter Eleanor. And we were able to use genetic variation to match up parent and offspring colonies. That is, we were able to figure out which colonies produced the daughter queens that founded the offspring colony. So what we're really measuring is the female component of reproductive success. Which offspring colonies were founded by daughter queens of which parent colonies? So this was amazing for me after all these years to be able to figure out who's related. This is another version of the map of my study site. This is from 2010, which is the year that we did this. And each symbol is a nest or a colony. Each symbol is a colony. And the size of the symbol shows how many offspring they had. And it's shown in two colors because there are two different genetic lineages in the population. You don't need to worry about that to understand this story. And this just shows the results for one family. So for example, from this work, I found out that colony 154, whom I've known well for many years, is a great-grandmother. Here is her daughter, her granddaughter, and her great-granddaughter colonies. Colony 154 happens to be a colony that I always thought was kind of a loser because on a really hot day, colony 154 would be there'd be maybe a trickle of foragers, where there were some other colonies out there, even some right nearby, that always have lots of foragers out no matter what. But in fact, it turns out that colony 154 is a great-grandmother. This shows all of the parent offspring colonies that we found in 258 colonies that were sampled in 2010. But these were colonies of many different ages, so because we knew all of their ages, we could go back year by year and ask, okay, of all the colonies that are 5 years or older in a given year and could be the parents, which are the parents of the offspring colonies produced in that year? And that's how we were able to find all of these parent-offspring relationships. By doing this, I could ask the question, is anything about colony behavior heritable? And it turns out that offspring colonies resemble parent colonies in their decisions about which days it's so hot that they don't forage. The offspring and the parents are so far from each other that the ants never meet. They're farther apart than they could possibly go in their foraging trails. So the offspring colonies can't be learning anything from the parent colonies. And this is why we'd like to look for the genetic variation that may underlie this resemblance, the apparent heritability of colony foraging behavior in parent and offspring colonies. So if there's genetic variation in parent and offspring colonies that explains why they resemble each other, it seems that it would have to be genetic variation in ant response to interactions. That is, maybe colonies in which each ant is more likely to forage only after it gets many interactions. Perhaps colonies in which the decay rate of an ant's response to interactions is very high, so that it takes a lot of interactions to accumulate enough to push it over the threshold to forage. Maybe a colony like that is likely to have offspring colonies that are also behaving that way. And this is what we're going to be looking for. So finally, the punchline. Is there a relationship between colony behavior and reproductive success? And we found that there is. So the colonies, like colony 154, that are more likely to conserve water and sacrifice getting as much food as possible by reducing foraging more on a really hot day, those are the colonies most likely to have offspring. So it's not the colonies that go out and get the most food, it's the colonies that save water. I think that the colonies that conserve water more have better hydrated queens who, when they leave for the mating aggregation and have to spend the first 6 weeks in their own nest before they produce the first workers who can go out to forage for them, they may have an edge on the queens from colonies that haven't been as good about saving water. Over the time of this study, and especially in the last 10 to 15 years, there's been a very severe and deepening drought in the southwestern US. And it may be that natural selection is acting this way in response to the drought. And it's certainly possible that in different environmental conditions, evolution will push the regulation of foraging in another direction. To my knowledge, this is the first time that we've been able to track the ongoing evolution of the regulation of collective behavior in a natural population of animals. What's happening is that more successful colonies conserve water by foraging less in poor, dry conditions. But when conditions are good and it's more humid, those same colonies actually have more stable foraging. So natural selection is acting to shape the way that ants are using local interactions to regulate the behavior of the colony. Interaction networks evolve in response to environmental challenges. By looking at how the regulation of collective behavior interacts with the environment, by asking ecological questions about collective behavior, we can learn how collective behavior evolves. Maybe we can learn from the ants, who give us beautiful examples of collective behavior in many different environments, how collective behavior is evolving in other environments and in other systems, like these cancer cells. Thank you.